Hi everyone, thank you for giving an opportunity to present. I'm going to talk about open modification proteogenomics that fosters reproducible detections of non-canonical proteins. As we all know, the traditional method for detecting proteins in complex samples is shotgun proteomics. And in a typical experiment, the database of annotated protein coding sequences is plugged into the search. And the identified proteins is the set of initial database with a couple of variable modifications. But when we add a comprehensive database informed by proteogenomics, we identified a bit more proteins, like non-canonical, and you heard a good introduction about them from Nicolas' talk. And when we do open search, it allows us to cover entire possibility of protein detections. This requires a robust analysis to achieve the least level of false positives. And it is possible under the assumption that the distribution of false positive and true positives is homogeneous. However, a target decoy approach is biased against the expanded search space. In proteogenomics, when small peptide subpopulations are introduced, the assumption about homogeneity is violated. In extreme scenario, when we combine proteogenomics and open search, there are even smaller groups like non-canonical peptides with rare modifications. And whilst FDR is underestimated, and what is thought to be under 1% FDR can actually correspond to 30%. However, can we do better with FDR control in open modification proteogenomics? Here we challenge search engine IronBot with this task. IronBot is semi-supervised machine learning engine that allows one unexpected modification per peptide. It narrows down the peptides candidate set by predicting sequence tags, and then rescores them with a semi-supervised model based on fragment features like predicted intensities and peptide retention time. We test IronBot on 3 databases of different size. The biggest one is OpenProt, the proteogenomics database with comprehensive set of non-canonical proteins. So we run IronBot on public datasets in an entrapment analysis to estimate false discovery proportion for every scenario, and we compared global and group FTR. In result, IronBot controls FDP under 2% despite the database size, with a slight advantage when we use group FTR. However, IronBot is also biased towards smaller peptide subgroups. As I mentioned in the beginning, the FDP of non-canonical peptides reaches 30%, while it is well maintained for canonical peptides. Group FDR applies more conservative filter on non-canonical peptides. However, it also creates inadequate FDP for canonical. Thus, we proceed with hybrid filtering, taking advantage of both FDR and which are based on peptide type. We compare IronBot to popular FragPipe tool with hybrid filtering, and identify that IronBot offers 30% gain in proteins identifications and 12% more PSMs and peptides per non-canonical protein. Open search increases the identification rate of closed search for 60%, which is expected, but also the most of the new spectra are matching the canonical peptides. Notably, if we zoom in here and looking on non-canonical peptides, we see that 40% of non-canonical PSMs are rematched to canonical from close to open search, and 0.5% of canonical PSMs are rematched to non-canonical in open search. The switch in annotation is justified by higher correlation of observed PSMs with predicted spectra. Thus, open search evidently increases accuracy of discoveries. What is striking is that 85% of non-canonical peptides are modified, comparing to 42% of canonical peptides. And non-canonical peptides also have a distinct modification profile. Like, for example, they're less often deamidated and more often have an oxidized proline. So we wondered, can the modification frequency influence what peptides we observe in our data? Non-canonical proteins are shorter, so the probability to detect N-terminal peptides is higher than N-terminal peptides of canonical proteins. But their relative abundance in open search is 10% higher than expected. So we investigated them more close, and it revealed that N-terminal peptides do tend to be modified more often than internal peptides, helping their detection in open search. Moreover, the modification pattern of N-terminal canonical and non-canonical peptides is closer to each other than internal. And this suggests that the modification characteristics can be more attributed to peptide localization in protein than to the protein group. Then we followed HPP guidelines to end up with the most confident set of non-canonical proteins. The bottleneck of the analysis was the filtering of the peptides that non-uniquely match to the single amino acid variants of canonical proteins. Then we also checked the peptides with mass shifts that are isobaric to single amino acid substitutions, and if they matched the known missense mutations, they were filtered out. Lastly, we also examined spectra manually and visually. However, I cannot tell you exactly how much of them end up good because we are continuing re-evaluation. But just for example, for your reference, this is the spectra of not very good coverage, where even the ions with good intensity predicted with MS2PEEP are lost. And for the reference, this is a better spectra with good quality and cleaner. These cases are quite trivial. However, this one is more complex, which I'm going to introduce. This PSM has a good coverage and comes from the peptide of pseudogenic protein. And if we compare its protein sequence to the parental gene protein through the alignment, we can find our peptide here. And it means that the identified pseudogenic peptide is just one amino acid different from the parental, exactly where the mass shift is found right here. And if we sum up the masses of threonine and the shift of the pseudogenic peptide, we end up with the glutamic acid, which is found in shared peptide. So now we are not really confident in this spectra, though it looks very good. To conclude, our study highlights the importance of open modification proteogenomics in discovering non-canonical proteins. 85% of them are modified, and it can potentially link their biological role. And our study also emphasizes that the manual spectral validation is crucial when we claim a new protein, even if we apply a stringent group FDR control. I would like to thank my colleagues, collaborators, and to organizers for such an amazing conference. Thank you. I'm afraid I don't hear anything if anyone's speaking.
Do you hear me?
Yes.
Okay, great. It was quite remarkable that FDR behaves so differently between like the canonical and non-canonical proteins. Do you have an hypothesis of why this behaves so differently?
Yeah, that's a great question. One of the reasons can be abundance, because the identification of non-canonical peptides we expect to be a rare event because they are shorter, they have less expression, and also DDA favors the more abundant proteins. So yes, in rare identifications we expect higher false positive rate. And what can be also linked to it is could be the spectral quality, and this is the ongoing work. So we'll make better conclusions if this can be linked or not.
Thank you.
Great. Thanks again, Valeriia.
Thank you.